Okay, yeah, with me, we are switching back to our favorite model plant, Arabidopsis, and also the talk will be a little bit smaller in scope than Allison's talk, but hopefully nevertheless interesting. So I'm going to talk about part of the work I did in Phil Wick's lab on temperature responses in plants. As you're probably all aware, temperatures like heat and cold have strong impact on plant physiology and plant yield. But what I'm mainly interested in is actually ambient temperatures, warm ambient temperatures, such as you may observe under warm summer days, and how they impact not on plant physiology, but on plant growth. And just see, there we go. So when you think about impacts of temperature on plant development, that is actually true for the whole plant life cycle. So we have we start with germination, which is sensitive to the temperature environment, through vegetative growth, be it elongation growth of shoots or stomatal development, and then through to reproductive growth. So the onset of flowering in many species is temperature regulated, as is fruit ripening and fruit abscission. However, the molecular foundations for these responses and for the sensing of temperature have only begun to emerge like in the last decade, and most of the work has been done in Arabidopsis. And when we look at temperature responses in Arabidopsis, a classical response we always look at is elongation of the hypocotyl, which you see depicted here. So you see if you grow your little Arabidopsis plant at 28 degrees, the hypocotyl, the embryonic stem, will dramatically increase in length. And that is a very classical response to look at. And in a seminal paper in 2009, it was found that the PIF4 mutant absolutely lacks this response. Now, PIF4 is a member of a transcription factor family called phytochrome interacting factors, and they had previously been well established as negative regulators of light signaling. However, over the last decade, it has become clear that PIF4 is also a central regulator of temperature-controlled growth, or thermomorphogenesis, as it became to be called. So it has become clear that PIF4 not only promotes hypocotyl elongation, but also petiole elongation, hyponastic growth of leaves. It represses stomatal development, and it promotes the onset of flowering in Arabidopsis. But the big question then became, how does temperature positively affect PIF4 activity? And over the last, let's say, 4 or 5 years, two temperature sensing mechanisms emerged. One involves the so-called evening complex, which is a protein complex made up by ELF3, ELF4, and LUX proteins. And just recently, it has been shown that ELF3 in itself is a bona fide temperature sensor through its prion-like domain. And this complex accumulates in the early evening, and binds to the PIF4 promoter and represses its transcription. Warm temperatures, however, reduce activity of this complex and thereby de-repress PIF4, so PIF4 transcript can actually accumulate during the night. But PIF4 is also regulated at the post-translational level by phytochromes. You probably all know phytochromes as red and far red light photoreceptors, but phytochrome B specifically also functions as a thermosensor in Arabidopsis. So during the day, most phytochrome is in its active PFR conformation because it absorbs red light, which activates it. But then in the night, it slowly reverts back to its inactive PR conformation. And that process can be accelerated by warm temperatures, which is why it has also now been called thermal reversion rather than dark reversion. So that means in warm nights, phytochrome B will inactivate much more quickly, which means by the end of the night, you have low levels of active Phy-B. PIF4, which is normally directly repressed and also degraded in response to phytochrome B, will become de-repressed. So we have active PIF4 at the end of the night, and that nicely correlates with an increased growth at the end of the night that we see in short days. However, if we think about like real life and warm temperatures in real life, they don't really happen in short days. So here I'm just showing you some empirical temperature data from 2 different summers in Cambridge. You can see in 2017, it wasn't the greatest of all summers, so temperature stayed pretty constant and barely scratched 20 degrees. 1 year later, we had quite a nice summer, so we got like nice cycling temperatures over the day, warmest temperatures observed at daytime with a peak in the late afternoon. And that is much more common for temperate plants to experience warm temperatures in this context. So we then decided to have a specific experimental setup to kind of mimic these conditions. So we grew plants in long days, which means 16 hours of light, 8 hours of dark, and either kept them at constant 17 degrees or gave them a short window of 8 hours at 27 degrees to mimic this warm phase in the day. And these experimental conditions have been used throughout the experiments I'm going to show you. And they're always indicated with these kind of boxes down here. So like the yellow box indicates the light phase, the black box, the night, and the red hatching here indicates the warm 8-hour window. So the first questions we wanted to answer is, do these conditions actually induce strong thermomorphogenesis phenotypes? And the simple answer was yes, they do. So we looked at our favorite hypocotyl elongation phenotype and saw that you get strong elongation under our cycling warm temperatures. So hypocotyl length essentially almost tripled in these seedlings. We were also able to observe seedlings over several days using an infrared camera. And you see the results of that here. So you see little seedlings start to grow at both temperatures, a bit slower at the lower temperature. But essentially, there's a burst of growth in the beginning at both temperatures. But then the seedlings at 17 degrees arrest, whereas those at 27 degrees keep on growing. And they keep on growing in a phased pattern. So they grow for a few hours and then stop, and then will resume growth in the next day. All right, just waiting for that to play out. When we quantify these growth rates over the time of day, we can see here with the red curve that growth happens almost exclusively during the day, and it actually peaks at the warm phase of the day indicated by red hatching. And this is quite distinct to the response that we see in short days, where, as I said, growth happens at the end of the night. So it really seems that there are at least two different pathways at work, one that controls temperature-dependent growth in short days, and one that controls temperature-dependent growth in long days, and that acts at daytime. We also checked other phenotypes and found that our conditions induce strong petiole elongation. And we also looked at flowering time, where our conditions only had a small accelerating effect, at least at 27 degrees. And only if you cranked up the temperature to 37 degrees, you actually got significantly accelerated flowering in Arabidopsis. Now, at this point, we were wondering what players could be involved in this response. And we were hypothesizing, because it seems to be a different pathway, that maybe it's not only transcriptional control that plays a role, but also translational control. The reason for that being is that translational mechanisms are known to gate growth in the context of the circadian clock and light signaling. So maybe that's happening with temperature as well. So at this point, we teamed up with Betty Chung, who is an expert on investigating translation in plants, and decided to have a global look at translation in response to temperature. So what Betty did is she grew seedlings in 17 degrees, then shifted part of them to 27 degrees for actually a very short amount of time, because we were interested on rather quick responses. And then she looked both at the translatome and the transcriptome of these seedlings. For the translatome, what you do is essentially you purify RNA fragments that are bound and thereby protected by ribosomes. And they will basically serve as a proxy for translation. So you quantify these ribosome protected fragments by next generation sequencing and thereby obtain the translatome of your seedling. And in parallel, Betty performed a standard RNA sequencing experiment to get the transcriptome as well. And then, because we were specifically interested in translation, what Betty did is to normalize the ribosome protected fragments by the number of RNA fragments to specifically only look at the translational effects. And this is exactly what is plotted here. So you see on the y-axis, the fold change in translation efficiency between 27 and 17 degrees. And that is plotted against the fold change in mRNA levels. So everything that's shifted along the y-axis shows clear changes in translation efficiency in response to temperature. And among these genes, we found PIF7 to be enhanced at the translational level. Now, PIF7 is quite interesting because obviously it's closely related to PIF4, which is the key regulator of thermomorphogenesis in Arabidopsis. But PIF7 at that point had not been implicated in temperature responses. So we decided to investigate PIF7 a bit further. And the first thing we wanted to confirm is that temperature actually has a positive effect on PIF7 protein levels. So we generated a PIF7 transgenic line to observe PIF7 levels by Western blot, which is exactly what you see here over a 24-hour day, and focus on the lower band here. And you will see that the bands we observe at 27 degrees always seem to be more intense than those at 17 degrees. And that becomes more clear when you look at the PIF7 protein quantification here on the bottom left. You see that the effect isn't dramatic, but we always get a clear, a robust, and a reproducible increase in PIF7 protein levels at 27 degrees to about 2 to 2.5 fold at the max. In contrast, when we look at transcript levels here on the bottom right, we don't detect any significant differences between the two temperatures, clearly showing that the effect temperature has is post-transcriptional. Now, at this point, we had to answer the question, is PIF7 actually involved in thermomorphogenesis in Arabidopsis? As I said, it hadn't been implicated in that before. We went back to our favorite Hypocotyl assays, and that gave us a very clear answer. Yes, so Hypocotyl elongation at warm temperature is dramatically reduced in the PIF7 mutant to almost the same extent as in the PIF4 mutant. And then when you look at the double mutant, you don't see any further decrease. So it seems that both PIF4 and PIF7 are quite essential for the temperature response, at least during daytime. We also investigated other thermomorphogenesis phenotypes. We found that the PIF7 mutant had a severe reduction in petiole elongation. And we also found that stomata development was affected. So while in the wild type, stomata development is reduced at warm temperatures, as you can see here, that effect was completely abolished in a PIF7 mutant. So it seems we get very clear phenotypes at by looking at plant growth. But what about at the molecular level? To address this, we performed an RNA-seq experiment, which I analyzed with the help of Fong in the lab. And in this RNA-seq experiment, it was basically a time course of wild type and PIF7 mutant plants grown at 17 and 27 degrees. And if we first focus on the upper panel here, you see that's the wild type compared at 27 and 17 degrees. Green colors indicating upregulated genes, magenta colors indicating downregulated genes. And you see there's a lot happening in both directions in response to temperature. However, in the lower panel, we are comparing the PIF7 mutant versus wild type at warm temperatures. And you see that panel is mostly white. So there isn't that many genes misregulated in that mutant. However, there was misregulation detected in one particular cluster, in cluster 7 here. And you see that these genes show a strong induction in the wild type at warm temperature, specifically in the middle of the day at ZT8 and ZT12, which is exactly when the plants experience our warm temperature treatment. And this upregulation was completely abolished in the PIF7 mutant, so these genes weren't induced at all. And when we performed GO-term analysis of this cluster, we basically found all GO-terms related to the phytohormone auxin, response to auxin, auxin signaling pathway, etc., etc. Now, this in itself wasn't that surprising because auxin is well established as an essential component of temperature-dependent elongation responses. But nevertheless, it confirmed that, PIF7 is really important for these elongation responses. In parallel with the RNA-seq, we also performed a Chipseek experiment with the help of Katja Jäger in the lab. So we used our PIF7 MIC line to look at directly bound genes by PIF7, which is indicated also up here with the blue sticks. So these represent genes directly bound by PIF7. You see there is a good number in cluster 7. And when you look at the binding intensity of PIF7 in our Chipseek, you see that the binding intensity increases at the warmer temperature, which is indicated here by the red and orange lines compared to the blue and purple lines. And that is essentially a consequence of the higher protein levels we observe under these conditions. We also looked at the intersection between genes bound by PIF7 and whose expression was dependent on PIF7. And we found the overlap was quite small. So there's a number of genes that are bound, but they don't show misregulation in the mutant. And there's a number of genes that are misregulated, but not directly bound by PIF7, but rather are indirect targets. However, among these putative direct targets, these 82, this blue group in the middle here, we found the Auxin biosynthesis genes YACA8 and YACA9. And you can see on the right here, both YACA8 and YACA9 show a nice peak in expression under warm long days in the wild type in the middle of the day. And this peak is completely gone in the PIF7 mutant. And on the left here, you can actually see the Chipseek profiles for the two promoters of these genes. You see that both promoters are bound by PIF7 and binding increases towards warmer temperatures. So clearly showing that these two genes are likely to be key regulators that act downstream of PIF7. To further investigate that, we looked at a YACA quadruple mutant as well as a YACA8 overexpressor line. And we found that the quadruple mutant essentially lacks the elongation response at warm temperature. So it looks very similar to a PIF7 mutant, whereas the overexpressor shows a long hypocotyl even at low temperature. We also investigated whether we can complement the PIF7 mutant phenotype by adding exogenous auxin. So we treated PIF7 seedlings with smaller or low amounts of the synthetic auxin picloram. As you can see here, 500 nanomolar picloram caused an elongation of the PIF7 mutant, so it could at least partially complement the PIF7 mutant phenotype, showing that auxin acts downstream of PIF7. So at this point, we had a pretty clear idea of what's going on molecularly downstream of PIF7, but what is happening upstream? So how could temperature possibly affect PIF7 translation? To tackle that question, we actually took a step back and looked at different systems where temperature-controlled regulatory mechanisms for translation are known, and this is mainly in prokaryotes. So here's a little scheme of translation initiation and how it happens in prokaryotes. Basically, the small ribosomal subunit will recognize the so-called Shine-Dalgarno sequence, which is close to the initiation codon. And then the whole functional ribosome will be assembled. So the tRNA and the large subunit will be assembled into a functional ribosome. And there are well-established mechanisms by which this process is regulated by temperature through so-called RNA thermometers. RNA thermometers are secondary structures that form across the Shine-Dalgarno sequence and restrict access and the ability to recognize it by the ribosome. And these secondary structures are temperature sensitive. So if temperature rises, they may either melt or adopt a distinct conformation, which essentially frees the Shine-Dalgarno sequence, which becomes accessible again and translation can occur. Now, if we go into eukaryotes like plants, the situation is a bit different. In eukaryotes, a so-called scanning complex assembles at the 5' end of the transcript, consisting of the small ribosomal subunit, the tRNA, and a number of initiation factors. And the scanning complex then scans along the 5' UTR of that transcript until it finds the initiation codon, whereupon the functional ribosome gets assembled. However, you could easily imagine that secondary structures could also interfere with the scanning process. So there's absolutely no reason to think that RNA thermometers couldn't exist in eukaryotes. Yet, at that point, no eukaryotic RNA thermometers were actually known. So we were intrigued by this and decided to investigate whether that was one of the mechanisms by which temperature affects PIF7 translation. So the first step we wanted to do was to look at potential secondary structures in the PIF7 5' UTR, and we used minimal free energy as a way to predict secondary structure formation. And we found that the most likely secondary structure in the UTR is actually a hairpin that forms rather close to the initiation codon, which is depicted here. So we decided to investigate the functional relevance of this hairpin, putative hairpin, I should say, at this point. So we took the 5' UTR of PIF7 and fused it to a luciferase reporter. And we not only used the wild type sequence, but we also mutated their sequence with the idea to have an impact on hairpin formation. So we mutated either side of the hairpin, which should disrupt hairpin formation altogether. We mutated both sides of the hairpin, which should then reconstitute the secondary structure. And we also introduced additional GC base pairing, which should stabilize that hairpin. And then we essentially performed in vitro translation assays. To do that, we transcribed the constructs in vitro and then used similar amounts of RNA for in vitro translation in a wheat germ extract system. And we incubated these mixes at different temperatures for a defined amount of time. Then we stopped translation by adding cyclohexamide and then measure luciferase activity as a proxy for the translation rate. And our idea was that, okay, maybe this hairpin functions similar to the bacterial or prokaryotic RNA thermometers. And by saying that, I mean that it will restrict translation at low temperatures. And if you break the hairpin, then you get high translation all the time at all temperatures. However, that was not what we saw. What we saw instead was that there was little translation happening at low temperatures, but there was a clear difference to be seen at warmer temperatures of 27 to 32 degrees. So we found that the wild type sequence, which is the line here in red, and the reconstituted hairpin sequence in orange showed highest luciferase activity and thus highest translation rates, which were clearly reduced in the disrupted hairpin sequences in blue here. And they were even further reduced in the stabilized hairpin. So it seems that the hairpin we identified in the PIF7 5' UTR acts as a translational enhancer rather than as an obstacle, and it acts as an enhancer specifically at warm temperatures. And it furthermore seems that a certain flexibility of that hairpin is required for it to have the positive effect, because that is essentially completely abolished when we stabilize that hairpin. At this point, we then decided to do some more biophysical characterization to make really sure that the sequence is actually capable of forming the hairpin. And to this end, we teamed up with Marco Di Antonio, who is now at Imperial College London. And the first experiment Marco did was to use CD spectroscopy to characterize our hairpin at 17 and 27 degrees. And what Marco got were classical CD spectra for an RNA hairpin. So you get a minimum around 210 nanometer and a maximum around 270 nanometer. But furthermore, what he also could observe is that the hairpin seems to form at both temperatures, but the hairpin isn't identical. It seemed that at 27 degrees, the hairpin was a bit more relaxed, so it adopts a slightly different conformation at this temperature. And he then decided to investigate this effect further using intramolecular FRET measurements. So in these measurements, you tag either side of your sequence of interest with a FRET fluorophore. And then if a tight hairpin is formed, these fluorophores will be very close together and you get high FRET efficiency. However, if that conformation changes and the hairpin relaxes, the distance between the fluorophores will increase and thus the FRET efficiency will drop. And this is exactly what Marco observed here. So the FRET efficiency at 17 degrees was around 0.3 and dropped by about a third at 27 degrees. And this effect was also highly reversible. Marco also performed this experiment over a range of temperatures and could see that this drop in FRET efficiency specifically happened between 22 and 27 degrees, which is in good agreement with our in vitro assays, but also with the phenotypic data that we obtained using the PIF7 mutant. So the last thing for us to do was then to investigate whether the hairpin sequence is actually relevant in Planta. So to this end, we introduc' the hairpin mutations we had used in vitro before into our PIF7 MYC transgenic construct. So we introduced mutations in the 5' UTR and then transformed the whole reporter line into the PIF7 mutant. And the first thing we analyzed in a number of independent transgenic lines was protein accumulation. I should more specifically say we looked at the fold change in PIF7 levels between 17 and 27 degrees. And while there was quite a bit of variation between the different independent lines, there was a trend that emerged. That was that the biggest fold change was observed with the wild type hairpin sequences or the reconstituted hairpin sequences. And we detected a significant reduction in fold change for at least one of the disrupted hairpin lines, the light blue one here, as well as the stabilized hairpin line in purple here. We then decided to also look at the phenotypic level, whether the hairpin has an effect. So we used the same lines that we used for protein accumulation to also look at hypocotyl length. And when you look at 17 degrees on the bottom left here, we see that essentially all seedlings remained short. And that was true no matter how much protein level was already present under these conditions. So even if you have like a line that has huge amounts of PIF7 protein, at 17 degrees they won't do much. So clearly there is another pathway acting here that prevents growth from happening. However, when we look at 27 degrees, we actually see a nice correlation between protein level and hypocotyl length. So while most lines, or almost all lines, essentially complemented the PIF7 mutant phenotype, the amounts of PIF7 protein determined how much of complementation you get, or how much of overcomplementation, I might say, you get. So clearly, PIF7 protein levels are relevant for the phenotype under warm temperatures. And in the final experiments, we also decided to look at the molecular level. To this end, we selected lines with the different hairpin mutations that had roughly equal PIF7 levels at 17 degrees. And then we looked at a number of target genes in these lines. So we chose YACA8, IAA19, and IA29. The latter two are two Auxin signaling genes. And all three genes are strongly impacted in the PIF7 mutant, so they essentially completely lack induction in a PIF7 mutant. And when we looked at their expression in our hairpin mutants, we found that at least for YACA8 and for IAA19, we saw a nice increase in their levels in the wild type line and also in the line with the reconstituted hairpin. But this induction was almost completely gone in the disrupted hairpin lines as well as the stabilized hairpin lines, again being in good agreement with in vitro data and also protein accumulation data. Right, that essentially brings me to our model of how temperature controls PIF7 function. So we have shown that there is a RNA secondary structure, an RNA hairpin formed in the PIF7 5' UTR. This hairpin is temperature sensitive and adopts a slightly more relaxed conformation at warm temperatures. And this relaxed conformation acts as a translational enhancer and has thereby a positive effect on PIF7 protein accumulation. Increased levels of PIF7 protein will then lead to increased target gene induction, such as YACA8, and increased Auxin biosynthesis and signaling essentially will lead to increased elongation growth. And finally, I also can put this model back into our scheme of different temperature sensing mechanisms. And obviously, the hairpin can sense temperature at any given time of day. But since PIF7 transcript accumulates mainly during the day, this whole pathway seems to be mostly active at daytime, and thereby it nicely complements the other pathways which act in the evening and at nighttime. Right, and obviously at the end, I would also like to thank some people. First of all, Phil Wick for giving me the opportunity to work on this very exciting project, all members of the Wick Lab who I have tried to acknowledge during the talk. I also want to thank Alexander Jones and my current lab for hosting me after Phil went back to Germany. Betty and Marco, our collaborators, as well as EMBO and the Gatsby Foundation for funding. Yeah, and I'm happy to take any questions.
Okay, thanks very much, Martin. Yeah, that was really great. Nice stepwise investigation through the function of PIF7 there and the thermosensor. So yeah, we have a few questions. So one from Barandagraph. He asks, did you exclude any microRNA interference directed towards the 5' UTR hairpin formations upon temperature change?
Oh, that's a really nice question. I haven't been asked before, actually. We can't exclude it. There are no known microRNA target site as well, as far as I'm aware, in the PIF7 5' UTR, but essentially we can't fully exclude that. And we do think that there might be, in addition to our hairpin, it's well possible that there are additional mechanisms by which temperature affects protein accumulation of PIF7.
Okay, so there's a question from Mirth Prath, maybe, and apologies for the pronunciation. So PIF7 protein shows a circadian rhythm. Did you check the possible involvement of post-translational modifications leading to the degradation and or stabilization of PIF7?
Yes, so we looked at stabilization of PIF7. So we did assays with cyclohexamide, which will block translation, and then looked at PIF7 levels over a time course, and we also looked at PIF7 levels under MG132 treatment, which basically blocks the proteasomal degradation. We didn't find any effect of proteasomal degradation that would be temperature specific, so that would not explain the increased PIF7 levels at a warm temperature. As far as other post-translational mechanisms goes, there is a paper out there saying that PIF7 is regulated by phosphorylation, but phosphorylation in this case would not affect the degradation of PIF7, but rather its subcellular localization and effects would essentially control exclusion of PIF7 from the nucleus. We haven't looked at that, and it's well possible that such a mechanism operates on top of our thermoswitch.
Yeah, absolutely. I do notice there's been quite a bit of publications on PIF7 over the last year or so, actually. So there's a question from Maria Piriri, and she asks, she has a question about the rest of your translatome, transcriptome data set. So have you checked it in more details, and do you see more of these predicted hairpin structures within your data set?
Yes, excellent question. We looked at that. There is no global trend. So the global trend is actually that you get higher translation when there's less structure in the 5' UTR, which actually is very common also in other species. However, there is a subset of transcription factors that have similar hairpins and are enhanced at the translational level. So we think that while it's not a global effect, there are more transcription factors out there that are regulated in a similar manner, and we actually tested two of them in vitro at least. That's WORKY22 and HSFA2, and here they seem to be regulated in a similar way, but we haven't checked relevance of this regulation in planta.
So they're regulated in a temperature-specific way, or just in
temperature-specific? Yes, so in a temperature-specific way, but they also have a similar hairpin as PIF7 has in its 5' UTR, and mutating that hairpin has a similar effect as it does in the PIF7 transcript.
Okay, so a final question from, oh, here we go. And actually, there's another question coming in. So Caroline McKenzie asks, do you know why the flexibility of PIF7 hairpin is essential?
Yeah, that's a really good question. We don't know that yet. So, and that plays into the question: how does actually a hairpin act as a translational enhancer rather than as an obstacle or repressor of translation? We have a few hypotheses out there. So it could be that the translation helps positioning the ribosome over the initiation codon, which relates to the fact that all these hairpins have a certain distance from the initiation codon. It's also possible that this conformation at warm temperature is recognized by an initiation factor or something like that, and then helps initiating the translation process. But we don't know that yet for certain.
Okay, right. So let us end by, let me ask a question about the relationship between different temperature sensors. So there's another high-profile publication from the Wiggy Lab recently about ELF3, and it has a prion-like domain that has some sort of control. So is the regulation of temperature through that mechanism just differs from the ELF7, as you said, a different time of day, or is there any interactions between these things?
I think there is interaction. If I recall correctly, ELF3 does not affect PIF7 transcription, but I do think it affects it post-translationally. So I do think there is interaction between the two proteins, but that has only been explored in the context of circadian clock and gating function, but not in context of temperature function, as far as I know.
Okay, I see. Okay, there's no doubt that it's a complicated system, as you'd expect, of course. So
absolutely, yeah.
Okay.